Hi, this video will explain about cofactor. So the learning outcome is for students to be able to define cofactor, explain three types of cofactors, and their functions, which involve metal ion activators, coenzyme, and prosthetic group. What is cofactor? Cofactor is a non-protein molecule or ion that is required for proper functioning of an enzyme. Cofactor can be permanently bound to the active site or may bind loosely and reversibly. Reversibly means they can attach and detach. So that is why it is said bind loosely with the substrate during catalysis. So cofactor either bound to the active site or bound to the substrate during catalysis. There are two terms that we usually use in terms of enzyme. Enzyme and inactive enzyme without cofactor is called apoenzyme. Once the cofactor bound to the active site, the enzyme is activated, so it is called holoenzyme. Okay, but basically we just use the word enzyme. But if you come across these two terms, you have to know that apoenzyme is the inactive form of an enzyme, and holoenzyme is the enzyme with the cofactor. How does cofactor work? First, it enables substrate to bind to the enzyme's active site. It also helps to carry atom or any chemical group from one substance to another. Cofactor also induces the active site's conformational change to enable substrate to bind. So it induces the change of the active site's shape so that substrate can bind, can fit perfectly into the active site. There are three types of cofactors. The first one is metal ion activators. Then we have the coenzymes, and the last one is the prosthetic groups. So let's look at how do they differ from each other. Metal ion activators are usually inorganic ion. So it acts as an activator that binds temporarily or loosely to the enzyme. Meanings after the process, the cofactors, the metal ion will detach from the enzyme. So the binding of the metal ion to the enzyme will change the conformation of the active site of the enzyme. So the active site becomes more compatible with the substrate. Let's look at this example. This is the enzyme, the apoenzyme. Then the metal binding site, the metal ion bind to the active site of the enzyme. Thus induce a change of the active site of the enzyme, which becomes more compatible with the substrate. Before the presence of the metal ion, or before the binding of the metal ion, the active site shape is not compatible to the substrates. An example of metal ions that act as cofactors are magnesium, zinc, ferrum, copper, and calcium. Calcium as the example of inorganic ion. Example in blood clotting, calcium ions is involved, which helps the prothrombin to be converted to thrombin, so the blood clotting occurs. The second type of cofactors is coenzyme. Coenzyme is an organic but It is a non-protein molecule. Metal ion is not organic, but coenzyme is an organic. But it is still not a protein. So it's usually involved. Coenzyme are usually mostly are vitamin or derivatives of vitamin. Derivatives maksudnya terbitan-terbitan daripada vitamin. Coenzyme binds loosely to the active sites of the enzyme. Act as carrier that transfer chemical groups, atoms or electrons from one substrate to another. It also acts as carrier of hydrogen, atoms, electrons of molecules. Examples of coenzyme include an AD+, which we will see more often in our next chapter, cellular respiration. Vitamin B, coenzyme A, and also ATP. An AD+ is a nicotinamide adenine dinucleotide. So it is a coenzyme for dehydrogenase enzyme that act as hydrogen acceptor. So an AD+ will be discussed in more details in the next chapter. These are lists of coenzyme derivatives of vitamins. The third types of cofactor is prosthetic group. Prosthetic groups is a non-protein molecules, of course, and organic. Different from the previous types, the coenzyme and the metal ion, prosthetic group binds tightly or permanently to the enzyme and becomes part of the enzyme. So it becomes part of the enzyme. It stays permanently, bound permanently. So this prosthetic group, this cofactor carry groups of atoms, single atoms, and electrons. Example is the heme group of our hemoglobin. FAD, FMN. These two molecules we will be discussed, or we will come across again in cellular respiration. So this is how prosthetic group, an example of a prosthetic group that bound permanently to the enzyme. So it prepares a binding site for substrate, a more compatible binding site. So cofactor, as a summary, cofactors are non-protein molecules or ion that is required for proper functioning of an enzyme. Metal ion binds temporarily or loosely to change the conformation of the active site of enzyme. Coenzyme also binds loosely and temporarily to active site of enzyme, while prosthetic groups binds tightly or permanently to the enzyme and becomes part of the enzyme. Is there any cofactor in our coenzyme? Why? Because if cofactor is there in a coenzyme, then a coenzyme will become holoenzyme. Okay, thank you. That's all for cofactors.